is a third-time presenter. So
First time.
he's a third-time presenter, so that means you guys can ask him as many challenging questions as you'd like. He is presenting on dogs and the microbiome. So give it up for Gaurav.
Okay, so the title of my article is House Dust Exposure Mediates Gut Microbiome Lactobacillus Enrichment and Airway Immune Defense Against Allergens and Virus Infection. Or, a concise summary of why, if you haven't already, you should go out and buy a dog right now. So, the problem. As mankind has progressed, we've started spending more and more of our time indoors, to the point where, according to the researchers who wrote this article, 92% of our time in Western civilization is spent indoors. And a result of this is that the indoor environments that we spend a lot of our time in are starting to have a greater impact on our immunity and our ability to fight diseases. So, scientists have known for a while now that the dust and bacteria that develop in your home and in your workplace have an impact on your personal health. Specifically, it has an impact on your gastrointestinal microbiome. So, what that is it's pretty much a cluster of bacteria, thousands of bacteria that live in your intestinal system, from your esophagus, from your stomach, through your intestines, and in your colon. And most people think that all bacteria is bad, but in reality, you have a lot of bacteria living in your intestinal system that's actually good for you. They help you, digest food and fight diseases. And the gastrointestinal bacteria specifically is very important. And the researchers who kind of wrote my article wanted to know whether or not the indoor environments, the bacteria that you have in your homes, had any impact on the bacteria that you find in your body. Any impact whatsoever, either positive or negative. But before I continue, I want to do a quick survey. So, if you have a doggy, right, stand up. Okay, now sit down sit down or keep standing actually, if you've had your dog for more than 5 years or more. Okay, people still standing. Raise your hand if, as well as having a dog for more than 5 years, you also have very bad seasonal allergies. Okay, good. So not many of the people who are standing have their hand. You can all sit down. Thank you. So the people who are standing that had their hands down, that didn't have seasonal allergies while also having a dog for a long time, is kind of the reason the researchers who wrote my article did what they did. So what they wanted to test was whether or not the dust found in houses where people have dogs, the dust that accumulates from having a dog, had any impact on your gastrointestinal microbiome. So what they did was they set up an experiment where they had 4 different groups. They had a control group. They had a group of mice. They used mice for in this experiment. They had a control group where they just had mice, regular mice. They had a group where they had mice who were given dust accumulated from cockroach exposure. So they took dust where they knew cockroaches had been and used that on the mice. They took dust from households where there were no pets, so just regular dust from households that didn't have any dogs. And then they took dust from households that had dogs. In this experiment, they didn't have a preference as to indoor or outdoor dogs. They used a combination. They just wanted to see whether or not the mice with dog dust had any difference in the bacterial development in their intestines. And before we go into what actually happened, I just want to point out what this is. The the Th2 cytokine cell is, all you have to really know about it is that it's important in allergic response. So if you have allergies and you go outside and you get an allergic reaction to pollen, for example, Th2 cytokines are somehow related. They're probably what's causing that reaction. They not only they constrict the airways, but then as you'll see later, something else also develops immune response as well. But the main purpose of the Th2 cytokine in this paper is that it causes allergies. Yes.
So they're gavaging, right? So they're putting a tube down into the stomach.
Yeah. So this means gavage or gavage, however you want to say it. That pretty much means force feeding. They're giving these mice dust. They're force feeding them these 4 dust samples.
But if this is a microbiome experiment, would the acid in the stomach destroy all the bacteria?
No, I don't know. Maybe. I didn't say anything about destruction of their bacteria of the dust. I think they just looked at the effects.
Okay.
I'm sorry. Yes.
And are the levels of dust that they expose the mice to typical for exposure in households?
Yes. They actually that's a very good question. They had they realized that they used, I think it was 4 times as much dog dust as they did others because they saw that the concentration of dust was greater, so they wanted to keep the levels the same. Any other questions before we continue? Okay. So the first results that they found was that dog dust reduces allergy response. So what they did was they fed them dog dust and they wanted to see their response, the allergic response that resulted. And what they found was that dogs who were mice who were exposed to dog dust showed a decreased level of Th2 cytokine response. That's what these two are. These are 2 different types of Th2 cytokines. And as you see, the mice who were exposed to dog dust showed a less increase, a fold increase. Pretty much is like a multiple, like a 10-fold increase, for example. That's what that means. So what they found was that mice who were exposed to dog dust showed less increase than both the control and the mice who were given non-pet dust. And that's what this is. This is Th2 cytokine. And then they also found similar results when they looked at GOB5, which is responsible for mucose accumulation. So so when you have like a runny nose, for example, you have like a lot of mucus buildup, and that's what GOB5 is responsible for. And they found that again, the mice who were given dog dust had a decreased increase in GOB5, which leads to a lower allergic response. Yes.
Over what period of time did they notice those trends for like the allergy, for the reducing the allergy response?
It's a good question actually. It's in my paper. I don't remember exactly. I can get back to it. Just come back. Any other questions? Yes.
I don't know if you said this already, but did they expose the mice to the dog dust multiple times, or just like the one time and then make?
No, I think it was the one time.
But does that really make sense to do that though? Because you need like repeated exposure. You often need repeated exposure to an allergen in order to.
Yes, that is true, but I think for what they were looking at like the immediate response in all of these, and they found that the being exposed to dog dust actually had more of an immediate response.
And they didn't do any experiments where they repeatedly exposed?
They did. I'll show you that later, but they did do this again. Any other questions? Okay. So that was allergic response. They also found that dog dust reduces immune response. So this similar idea. They had the 4 groups, and they wanted to see the difference in the levels. And they found that this graph kind of it depicts lymph nodes, which cause lymph nodes are what's responsible for throat pain and swelling. And they found that against the control, all the mice were given dust in general, whether it was cockroach dust from non-pet houses or dog dust houses with dogs. They found that there was an increase in the number of lymph nodes, but they saw that between houses with dogs and not without dogs, they found that there was a less immune response in the mice who were given dog dust as opposed to non-dog dust. Now that was pretty much a significant portion of this graph.
So the immune response that it's reducing is, like causing the throat pain and all that stuff, and that's in reaction to the Th2 cells. Or
No, lymph nodes are separate from that. I think they were being triggered by T cells. T cells are what determine your immune response.
and we have not already determined that T cells were reduced by the dog test, or was that?
No, that was a Th2 cytokine that were used.
Okay.
Is dog dust like dog dandruff, or is it like the dust that you would have in a home with pets?
It's the dust you would have in a home. Now, they didn't take dog hair. And then the other thing that they noticed, in addition to lymph nodes, they looked at immunoglobulin response. They knew that immunoglobulin response causes inflammation, swelling, or bringing you into hives, which is not fun. And they wanted to see whether or not they had being exposed to dog dust had a increase or decrease in your immunoglobulin levels. And they found that, again, as consistent with the lymph nodes, they found that they found that cockroach dust and non-pet dust actually increased the number of immunoglobulin response compared to the control. But being exposed to dog dust kept it more or less the same. I think it's statistically insignificant that they're almost that this is a little bit less. I think the important thing is that being exposed to dog dust actually keeps your it doesn't affect your immunoglobulin response as compared to cockroach dust from not having it. Yes.
So in addition to the biological material for the dog, this dog dust is also containing a lot of the same materials in the no-pet cockroach dust, right? Because it's from household.
Yeah.
So, something about dog presence actually neutralizes the effect of those other dust types?
Very good observation. That's what we're going to determine later on. Yes, you're right. You'll see later on that they did find the presence of a dog actually did stimulate certain bacteria in your gastrointestinal system. Spoilers, by the way. Your gastrointestinal system, which positively affected the mice. Okay, yeah. Good question.
So why is it dogs? Why is it not maybe giraffes or
you don't have giraffes in your house. Yes, and I go back to the very first thing we were talking about was that they want to see your home environment, the environment of every person's home. How it affects your gastrointestinal system. So a lot of people, as you saw, most people split up. So a lot of people have dogs. So to see whether or not having a dog, which is something that most people have, had any impact on your home environment, the gut bacteria.
I think he's asking why versus cats.
Because no one likes cats.
So I don't the answer is no.
I don't know why they take dogs over cats. Because they didn't specify that. But I mean, I think more people have dogs than cats. Yes. Short answer is I don't know. Okay, we gotta move on. So they wanted to look at as they noticed the same thing. Was that they noticed that somehow having the dust of a dog was benefiting the the bacterial response, the immune response and allergic response of these mice, and they wanted to find out why. So they made this horrifying chart that you really don't have to know anything about besides this. They found that there was an increased level, an enriched level of lactobacillus johnson. And researchers had known previously that L. johnsoni was a gut bacteria that's generally considered a protective species, which means that it's beneficial to mammals, specifically in preventing respiratory syncytial virus. RSV is something that affects infants and can cause asthma later on. If you get RSV as a child, then there's a chance you'll have asthma later on. And so they wanted to see whether or not the stimulation of L. johnsoni had was related to the dog dust presence and had any impact of its own.
It seems like the Latin spiritae had a higher level of enrichment in the Microbiome johnson. Is there any particular reason that they chose?
There is, and it's not a very scientific reason. It's that the researchers who wrote this knew about L. johnsoni more than they knew about everything else, so they just moved down with it. Okay, moving on. So they did a similar thing as what they would like. Going back to what Liz's question was, whether or not they repeated this. The experiment they did, but this time they only used cockroach dust because cockroach dust is something that had been established before their paper, so it was kind of a control in this sense. And they wanted to see whether or not the presence of L. johnsoni had any impact on these Th2 cytokine cells from before, the allergy stimulating cells. And what they found was that, so the gray bars is protein expression, so I'm not going to really talk about that much. But what they found with the black bars is that the level of the mRNA level of cockroach, the level of C2 cytokine cells with cockroach dust plus this L. johnsoni significantly decreased, I might add, the amount of Th2 cytokine cells that were present as opposed to just the cockroach dust. So what they're able to conclude from this is that, L. johnsoni has a positive impact in reducing allergies. So it probably has some role in what's happening when you ingest dog dust. And then they also, going back to what I said before, they wanted to see whether or not it had any impact on asthma, because they wanted to look at respiratory syncytial virus, and they wanted to see whether or not L. johnsoni had any effect on that. So what they did was they did what you usually do when humans, you and I, go to get an asthma test. They expose you to methacholine, which kind of restricts your airways, and they want to see what the impact is, like how, if you could still, breathe well or not when you have exposure to methacholine. And so they did the same thing. They took cockroach dust and cockroach dust plus L. johnsoni. I wanted to see the difference, if there's any difference in the pressure. CmH2 per milliliters per second, if I got that right, I looked it up online. I think it's pressure. So I wanted to see the pressure on your airways, and they found that when given methacholine, L. johnsoni reduces the contraction of your airways, so it makes it easier to breathe, which in turn indicates a lower asthma response. Any questions? Okay. And so they determined that L. johnsoni had a beneficial impact on your immune and allergy response. But they wanted to determine whether or not they needed active or dead L. johnsoni. And between you, me, and the wall, I don't know why they did this, because bacteria really isn't effective if it's dead. But they did it anyway, and they found that, what is this? They found that you did need live L. johnsoni to have the beneficial effects. Hey, any questions on this? This is not that.
Aren't they looking for bacterial transformation? Isn't that why you're doing an active versus killed comparison? They're looking to see if there's going to be bacterial transformation, right?
Oh yeah, maybe. I didn't think of it. Yeah, okay, I guess they did have to look this up. I know. Anyway, they did find that you need live L. johnsoni. As you can see, the black bars are, the V, LJ means viable, L. johnsoni, H3 means heat killed. And you can see the heat-killed one wasn't very effective. You needed viable Allergies.
I think that the heat killed would also test for whether the effect was due to something on the surface of the bacteria.
I agree.
Thank you.
Okay, any other questions? Yes?
For the previous slide, at what point did they measure, like, what was the time period that they measured if it had any effect? Was it like 5 days after they put dust in?
I don't think it was 5, I think it was like, I don't know for sure, but, you know, I had to get an asthma test like a couple years ago, and it's like, you know, 10 minutes. From my experience, but I can it's probably in the paper. I can look at it and get back to you. Okay, so the conclusion, what they found was that so first of all, dog dust stimulates protective gastrointestinal bacteria. So pretty much, you know, being exposed to a dog it reduces allergy and immune response. It has a beneficial impact on your gastrointestinal microbiome, which leads to the next point, that infant mice and humans, by extension, exposed to dogs in their infancy, are less likely to develop allergies. So if you have a baby who's exposed to these different dog dust samples in their home from a young age, then they're less likely to develop the allergies that we saw before. They're less likely to have increased Th2 cytokine responses to, you know, certain like peanuts, for example, or pollen.
You said sorry, the infant mice are less likely to develop allergies, but aren't the studies showing like direct response? So like even if your kid is like 10 and you bring a puppy into the household, that will decrease their like C2 things.
Yes and no. They did find that, but I think the difference between mice and the difference between mice and humans is that humans kind of, you know, infancy is the best time to develop what?
I don't think you can make that distinction. Yes.
Yeah. Okay. I don't know.
Sorry.
Yes.
The different breeds of dogs affected like hypoallergenic versus like normal dogs?
They didn't test that. All they said was that they had 5 different households where they took the samples from, and all it said that they were indoor or outdoor. They didn't say anything. So what the big picture here? What does this mean? This means that the bacteria that you find in your home has a significant impact on the bacteria that you find in your body, which in turn has an impact on your immune and allergic responses. So, you know, your environment plays an impact on your ability to fight, to suppress allergies and possibly even fight diseases. So basically, to quote Mr. D, it's the next epigenetics. It's similar. Your environment has an impact on you, which is what epigenetics is all about. So that's it. That's my presentation. I'd like to thank these people.
I will be posting a link to this presentation on Liquid Bio if anybody wants it. I'll see you guys all at the exhibit on Saturday.